Thank you everyone for joining our webinar. I'm really excited to have Dr. Riham Ayoubi here with us today. She'll be discussing how you can characterize an antibody in your own lab. And I think this is a really important topic to really cover considering, the reproducibility crisis and the fact that dirty reagents can really affect your data and ultimately the conclusions that you draw from your findings. So Dr. Ayoubi will be sharing with us, some best practices for helping ensure that the reagents that you are using in your experiments are top quality and can give you reproducible results. So before I pass it off to Dr. Ayoubi, I just want to share a couple slides to introduce Protein Tech to you in case you are unfamiliar with who we are. So Protein Tech is a leading manufacturer of antibodies and immunoassays. We have a really large portfolio of antibodies that encompass many different research areas. And we also offer immunoassays like ELISA kits and IHC kits. And we also offer antibody labeling kits and cell separation kits as well. Within our Protein Tech brand, we also have our humankind branded cytokines and growth factors. And all our proteins are expressed in human HEC293 cells, so you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins. We also have our Chromatec branded nanobody-based reagents. And nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, which makes them ideal for immunoprecipitation, super resolution microscopy, and protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics. And Protein Tech Genomics manufactures antibodies conjugated to oligonucleotides for single cell RNA sequencing. As a company that's driven by scientists, we are also here to support our researchers. So our high quality and award-winning products are self-manufactured, and we offer the largest in-house portfolio of antibodies for the human proteome. We also know the importance of product validation and generating clean antibodies. So whenever possible, we validate our antibodies using knockout knockdown models. And also, as antibody experts, we have in-depth knowledge of immunoassay applications, and we can offer outstanding PhD level technical support. So with that, I'd like to go ahead and introduce our speaker for today, Dr. Ayoubi. So Dr. Ayoubi is a cellular and molecular biologist by training. She completed her PhD at the University of Montreal, where she studied membrane trafficking and signaling pathways. She then joined the YCharOS initiative as a postdoc at the NeuroSGC. And then YCharOS is an open collaboration with 12 antibody manufacturers and 2 knockout cell line providers with the mission to characterize commercial antibodies for all human proteins. Dr. Ayoubi has developed and scaled up the current standard procedures and coordinates YCharOS studies. So with that, I'll go ahead and pass it off to you, Riham.
Thank you, Afrida, and thank you everyone for joining us today. Just a second. So after that introduction, I will start my presentation. So today I will present the YCharOS project, Antibody Characterization through Open Science. I will share our discoveries after quantifying antibody performance. I will just put up my laser pointer. I will highlight the importance of knockout and knockdown characterization and the importance of including knockout controls in our testing. And finally, I will also share some tips on how to choose and test an antibody. When it comes to commercial antibodies, I think we can all acknowledge that there is a general struggle that I will briefly summarize here. One, there are too many products to choose from, and we all have our own habits when selecting, but still may not be 100% confident when purchasing a product. This eventually leads to purchasing multiple reagents and spending way too much. In fact, in the US alone, there is an estimated loss of $1 billion every year on commercial antibodies. In addition to that, time and samples and other resources are also wasted in the process. The issue arises from two main facts. The first one is that antibodies may perform differently depending on testing conditions, making them very unpredictable. And the second one is the absence of standard procedures to follow to ensure data reproducibility. And that's applicable to both academics and sometimes in the industry as well. This is why we started the YCharOS effort with the mission to identify top-performing antibodies for the whole community. Commercial antibodies are compared side by side in the presence of isogenic knockout controls. To obtain these knockouts, human cancer cell lines, in our case, or other models, can be genetically modified with CRISPR-Cas9, where the genomic DNA is targeted by an RNA complementary to the gene of interest and will guide the nuclease Cas9 to break the DNA double strands. This damage may be repaired by the cell, but in most cases still disrupts the expression of a protein. Translation of a protein can also be interrupted by interference with messenger RNAs. Double-strand RNAs are transfected into the cells, and dicer, which is also a nuclease, cleaves it into smaller siRNA pieces that are complementary to your messenger RNA of interest. The RISC complex guides this binding and eventually cleaves the messenger RNA, preventing its translation. YCharOS testing is conducted, as I mentioned, in human cancer cell lines in three major immunoassays: Western blotting, immunoprecipitation, and immunofluorescence. This is the case for our current testing, and we are expanding, and I will touch on that a bit later in the presentation. Another aspect of YCharOS's efforts is also the focus on renewable antibodies. There are three main types of antibodies: polyclonals, which are collected from immunized animals and are the product of multiple B-cell lineages, allowing it to recognize multiple epitopes on an antigen. But because of the production process involves different animal from batch to batch, these products are known to show variability from lot to lot. The second type represents monoclonals. They recognize one epitope on an antigen because they are the product of one single B-cell lineage that's transformed into an immortalized hybridoma to sustain production. While these reagents are considered renewable and provide a consistent supply of antibodies, hybridomas may suffer from genetic drifting over time. This is why the third type of antibodies is emerging as a solution. Recombinants are animal-free products that result from sequencing and cloning of the theoretical antibody gene and can be produced indefinitely while maintaining batch-to-batch consistency. While recombinants are typically a version of monoclonals, manufacturers are now investigating the success of actually combining multiple monoclonals that can then target different epitopes of the protein while maintaining that sensitivity of detection, but replacing the inconsistent traditional animal-derived polyclonals. Now that we clarified why we focus on renewables, let's go back to the YCharOS structure. To be able to test antibodies from multiple sources, We partnered with leading antibody manufacturers who agreed to send us their products in kind. We are also supported by these granting agencies right here that nominate and fund antibody testing for their proteins of interest. Each funder is focused on proteins related to a disease area, which has been mostly, in our case, the field of neuroscience. However, the YCharOS projects are now extending to other broader areas of research as well. I will now show an example of the YCharOS workflow. Synaptotagmin-1 was nominated by the NIH as part of the AMP-AD project, Accelerating Medicine's Partnership Program for Alzheimer's Disease. When a commercial knockout line is available with acceptable expression levels, then we use a commercial knockout. However, in this case, and based on DepMap's transcriptomics, a custom knockout was necessary in this cell line background HTT 116, for example. And this custom knockout was made in partnership with the Early Drug Discovery Unit at the Montreal Neurological Institute, where we are based right now. Antibodies are sent by the partners and are shown here in this table. This table is simplified. You will find many more details about each product on the actual report, which I will mention a bit later on. But I would like to clarify that a single star here indicates a monoclonal product, and double stars indicate a recombinant. In preparation for protein extraction, of course, to start with the testing, the target is first searched on UniProt, looking for information like theoretical mass and subcellular location. And in this case, because synaptotagmin-1 is intracellular, lysates were prepared and processed by SDS page, and the antibody screening in Western blot is done first. Antibodies catalog numbers are shown at the top of each corresponding blot and presented in alphabetical order. The chemiluminescent signal is shown at the top with each corresponding point staining, which is presented as a loading and quality control. Any signal that's detected in the wild-type sample but disappears in the knockout is considered a signal corresponding to the target protein, in this case, synaptotagmin-1. However, a signal that appears in both wild-type and knockout cells does not correspond to the protein. Next, the antibodies are tested for their ability to capture the protein in immunoprecipitation. For this, lysates from wild-type cells are prepared in a non-denaturing buffer and incubated with the antibody, then processed by SDS page. 4% starting material is run side by side with 4% of the unbound fraction, and of course the whole immunoprecipitate. Unbound fractions are the lysates that were not captured by the antibody in the IP and inform us about efficiency of the immunoprecipitation. For example, this antibody successfully captured synaptotagmin-1 in the IP, depleting it from the unbound fraction. But this antibody, for example, did not capture synaptotagmin-1, and it remained in the unbound fraction. And as compared to the starting material, you can also notice the enrichment of synaptotagmin 1 for further analysis like mass spectrometry. Then we move on to the immunofluorescence application. Wild-type and knockout cells are plated in the same wells to remove bias during imaging and analysis. The two cell populations are labeled with two different fluorescent dyes, but those channels are not shown here. The cells are rather highlighted in green when they are wild-type and in magenta when they are knockouts. The antibody staining channel is shown at the top, and the DAPI nuclear staining is shown at the bottom. This antibody, for example, recognizes synaptotagmin only in the wild-type cells. This signal disappears in the knockout, confirming the specificity of the antibody towards synaptotagmin 1. And of course, any signal that remains in the knockout then does not represent synaptotagmin 1. We are slowly incorporating flow cytometry testing in our reports. So for this protein, our team in the UK also tested these antibodies in flow cytometry. Similarly to the IF procedure, wild-type and knockout cells are also labeled with two different fluorescent dyes. Staining of wild-type cells is shown in green and knockouts in purple. The dotted histogram that you can see here represents secondary-only channel. Some antibodies, like this one here, for example, shows the same signal intensity in wild-type and knockout cells, which you can see overlapping here, and therefore is not suitable for flow cytometry. On the other hand, this antibody shows specificity towards synaptotagmin 1, where the signal is distinct from the knockout population that rather overlaps with the secondary-only channel, making it suitable for this application. Sorry. All results are then collected and presented in a report that's openly shared with the scientific community. All reports are uploaded to an open access data repository named Zenodo. Some are also submitted to F1000 as data notes for peer review and PubMed indexation. When applicable, our data is also incorporated in the product page of the tested antibody on the antibodyregistry.org and on the antibody search engine SciDab, which I will mention a little bit later on in the presentation. In 2023, we gathered all our data from testing 614 antibodies against 65 proteins, and we quantified performance. When products were recommended for the corresponding application, we found that 79% of antibodies succeed in Western blot, 58% in IP, and 39% in IF. When products were not originally recommended by the manufacturer, still half of the reagent succeeded and passed the knockout test in our hands in Western blot. 37% of the reagents still immunoprecipitated the protein, and 26% also immunostained the protein. It's worth noting that with our current numbers for more than 100 targets, the antibody performance still follows very much the same trend in all applications. When we separated performance based on antibody type, which I presented earlier on in this presentation, We noticed that recombinants often pass the knockout test and have a higher success rate as compared to other types of antibodies and across all applications. For example, in Western blot, 67% of recombinants that we tested were successful, 54% in IP as well, and 48% in IF, allowing us to conclude that recombinant antibodies are top performers. We then wanted to compare the results between knockout testing and another commonly used antibody validation technique. First, when we compared the percentage of antibodies that passed our knockout test, but were also knockout and knockdown validated by the vendor previously, we found that the results were very similar. 90% passed the Western blot test in our hands, and I already mentioned in Western blot, and 80% also passed the test in IF. On the other hand, when we compared our knockout results to orthogonal validation by the vendor, only 38% of reagents passed the YCharOS knockout test in IF, and 80% still passed the Western blot testing. I will clarify what orthogonal testing is. Orthogonal testing correlates results between an antibody-independent assay and signal from an antibody-based assay across multiple samples. Here's an example that shows, unfortunately, that orthogonal approaches may be sometimes misleading. This is a Western blot against a protein named GPNMB. And the Western blot signal is compared to RNA values, again extracted from the DepMap portal. Notice that the A172 cell line expresses the protein with 5.2 transcripts per million, with very low signal in Western blot. But in DMS53, there is barely any noticeable signal, although the RNA value is higher or similar at 5.6. This lack of correlation is also noticeable when comparing to shotgun proteomics, which DepMap also offer for a small subset of cancer cell lines. And you can notice that U87 cell line shows an intense signal for GPNMB with a proteomics value of 2.5. However, the A172 cell line shows a lower Western blot signal, although the proteomic value is higher at 3.8. So based on this analysis and this lack of correlation between Western blot antibody-dependent results and transcriptomics and shotgun proteomics independent of antibodies, this antibody could have failed the orthogonal testing and could have been overlooked. However, it actually shows no signal in the knockout when we use a U97 cell line, for example, and this knockout test confirms a specificity toward GPNMB. And I would like to mention that the opposite can also happen, where antibody can pass the orthogonal correlation, but ends up failing the knockout test. YCharOS's working model benefits the whole community, where all results from these different resources and consumables contributed for this mission are at your disposal at a community, since we are openly sharing them without eliminating any negative results. The success rate of the YCharOS approach is also high. For every project that we worked on, at least one suitable antibody is found for each application and for each target. This is an example for one of the very first projects that we worked on called ALS-RAP. And you can see that, for example, in Western blot, we were able to identify at least one product for 30 out of 32 targets, 25 out of 31 targets in IP, and 21 out of 29 targets in IF. And the results are similar also for other projects like TreatAD and AMPAD, for example. While a collection of 106 reports is available to help guide the scientists in choosing the best tool, our collection is still limited. However, we can still share what we learned from this subset of targets. This is why I will now point out a few important aspects for your consideration when choosing and working with antibodies against your protein of interest. So first, to select an antibody, using an antibody search engine like SciTab is quite useful, or other search engines are also available online. So here, for example, I search for my target of interest. You can search by gene or protein name, or even by UniProt ID. And after searching for an anti-TDP43, a protein highly involved in ALS, you can see that the results returned with an overwhelming 1,000 products. So to refine the search and for reproducible results, recombinants with genetic validations were my preference here. And after applying a couple more filters like the needed applications, here I chose Western blotting and IF, for example, I was left with 11 options. I would like to point out that when antibodies are also tested by YCharOS, this can be found under the accreditation portion here under refinement of the results as well. So now these 11 results were ready for my evaluation. A product can actually be knockout and knockdown validated based on manufacturer data sheet or on the literature, or both. In this case, this recombinant from Protein Tech, for example, was supported by knockdown validation internally on the data sheet, as you can see here, with all its, in addition to all its recommendations that you can scroll through. And it was also shown in a knockdown from a publication. And this is why SciTab can be helpful in this case, because it can narrow down the images for you. You can even filter further to see the exact image in the publication where the knockdown is shown. Another feature of SciTab, which I find sometimes useful as well, is that they highlight the actual number of citations for each product, which ends up being a final element to make a choice, usually when you're left with too many options, even after refining your results. So in our experience, a successful immunoassay depends on an effective and preferably renewable reagent that is, of course, consistent. And this is the very first thing we want to look for. However, for the reagent to be effective, we need to evaluate the data, as I mentioned earlier, and actually even your own data. So when working with a new antibody and you need to confirm its specificity because you found insufficient information online, knockdowns are an easy option, usually easily accessible, and can be easily optimized as well until you get a definitive answer. So for example, this knockdown may be optimized. If you're not very confident about it, you can opt for something that looks a little bit more convincing for you, and also that would be hard to dispute by others, where we see the reduction in the corresponding signal is significant. So data reproducibility is also part of the key, and that applies to reproducing your own experiments over time and also between users. And that highly depends on renewable reagents. Additionally, for a reagent to be effective, it must be suitable for the assay and, of course, reactive with the sample species that you are using. And to keep in mind that, as I shown in our statistics, some applications were not tested by the manufacturer. However, the antibody may still be investigated for its suitability for a certain application. Another key to successful antibody-based assays is the use of appropriate samples. And first, I would like to highlight adequate protein expression. Here's an example where anti-PLC gamma-2 detects a higher level of the protein in the THP1 cells compared to HAP1 cells, while the protein levels are only 4 times higher in the PMA-treated THP1 cells. This increase in expression made a tremendous difference when immunostaining. The same antibody was tested under the same conditions, following the same procedure, but in 2 different cell types. And it shows how the antibody performance was affected. In the lower expressing cell line, the antibody stained both wild type and knockout cells similarly, not identically, but similarly, showing a lack of specificity. But when tested in the higher expressing cell line THP1, the antibody now recognizes PLC gamma with absolutely no signal left in the knockout. Inadequate protein expression can also have a devastating effect on antibody using and antibody screening in general in our context, for example, in Western blot. So this is a CD44 antibody screening in HAP1 cells that express 2.6 transcripts per million of CD44. And you can, I counted this for you. It's only 3 antibodies that can detect the protein in Western blot out of 23 that are tested. However, when the same screening was done in the breast cancer cell line MDAMB231 with an RNA score of 9.6, now 17 out of the 23 antibodies can detect the protein, proving the importance of using appropriate samples with antibodies. Not only the sample source is important, but also the type of sample that you are using. For an intracellular protein, for example, an antibody will detect the protein in the lysate, but not in the extracellular medium. But on the other hand, for a secreted protein, the antibody will not detect the protein in the lysate. However, it will capture the signal in the extracellular medium, culture medium. The third key to success is following reproducible, robust procedure. Sorry. So for Western blot and IP, using appropriate protein extraction buffers and fresh protease and phosphatase inhibitors is crucial. For immunostaining, applying relevant permeabilization, for example, is very important for immunohistochemistry relevant antigen retrieval. Even when preparing a sample for Western blot, this one simple step, the nature of the protein must be taken into consideration. For a GPCR like S1PR1, for example, boiling prior to SDS page can result in aggregation, which you can see at the top here, and loss of signal at the corresponding molecular weight of the protein, but the signal can be easily recovered by heating or gently warming up the protein. In other cases, you do need to boil the samples to resolve an extreme complex, for example. And finally, the keyword that I've been mentioning since the beginning of this presentation, the presence of a knockout or knockdown control in the same cellular background as the parent is highly, highly recommended when possible. This confirms the reagent specificity in all the assays that you are using, but is also a model to study your protein of interest, the function of that protein. Making antibodies is a liability, but so is using them. While all 4 keys are in the hands of the manufacturer, notice that 3 of these keys are also in our hands as scientists and antibody users. Providers, of course, need to make sure they deliver the best quality products at the production and quality control levels, but testing them appropriately is then also on us as users to help have conclusive results and share any additional validation actually in publications. Going back to the robust procedures and the importance of that, even for an effort like YCharOS, all of YCharOS's efforts wouldn't have been possible without consensus procedures approved and supported by all the industry partners in bold here. With all that mentioned, YCharOS has a lot of strength, but also limitations, like the need to make a knockout for every protein we work on. There is the challenge of accessing cells with high endogenous expression, knowing that some proteins are very tissue specific and may not be expressed in cancer cell lines at steady state levels. This limitation may be sometimes overcome and can be resolved by using iPSC cells or inducing expression with cell treatment. With that, I will thank everyone who contributed to our studies and made this project possible. And thank you everyone for joining us today and for listening.
Great. Thank you so much, Riham. That was a very interesting talk. It's really interesting to see the process that, YCharOS goes through for characterization. And it's really such a great initiative. And, us as Protein Tech, we're really proud to have a partnership with you guys and, to be transparent about our products and kind of their performance. So thank you so much for sharing all that.
Thank you, Afrida.
We do have a few questions for you. The first question is asking, so an issue I often encounter during Western blot is the presence of numerous non-specific bands that are not mentioned on the supplier's website. Do you have any advice on how to avoid these non-specific bands, which can be problematic when analyzing Western blot? And they are specifically using the cell line MDA MB231.
Okay. So antibodies cross-react. It's just an inherent problems depending on the sample that you are using. the performance of the antibodies in a Western blot specifically may look very different between different cell types. So if you are able, I would say, to change the cell type, if you can study your proteins in another breast cancer related cancer cell line other than MDA and B231, maybe with a higher expression that allows the antibody to pick up the signal that's specific to your protein and interest, and all the noise signal that you're detecting right now may disappear. However, so this is part of it. Another part can be doing some fine tuning and some optimization of your actual blocking steps, because that noise can be eliminated sometimes with that, or using, I don't know if you are using BSA to dilute your primary antibody. Maybe you could switch to milk, which tends to have a little bit more power in blocking noise. So trying to optimize a little bit of your Western blot procedure. I know that another trick that also helps for me when possible is increasing the actual amount of protein you're loading onto the gel. But sometimes it can create more noise, so you have to just try and see if it can help.
Yeah, and then kind of a follow-up, they were asking, could they change the dilution of the antibody in the Western blot?
So all these changes in changing the small parameters that are related to primary, secondary antibodies, and even the amount of protein loaded is actually a good recipe for spiraling, I have to be honest. I would say the key is using a better, more ideal, more adequate sample where the antibody will pick up only the signal from your protein. Or another option would also be to change your reagent. If you have an option to change the actual primary antibody that you're using, you may have better results with that. I hope that helps.
Thank you so much. The next question is a question about one of your slides. So it says on slide 39, how long was the denaturation each? I used to boil the samples at 95 degrees Celsius for 5 minutes in SDS loading dye. On this slide, it seems to already be aggregating.
Yes, so in this case, it was a 10-minute boiling. So actually, the standard procedure here is to like heat. Our standard procedure is to heat up at the 65 degrees for 10 minutes for the sake of showing what could go wrong with the GPCR. I did boil for 10 minutes. So I think heating up at 65 can be a good start, and then you can optimize from there depending on the protein you're working on.
Great. Thank you. The next question is, someone said that they attended Carl's talk at IRIC and looked out at your papers, but just considering the Western blot themselves, do you think the unique protocol you use is suitable for everything? So including using only, one type of membrane, one type of blocking, or one type of lysis buffer?
The answer is I can't answer for every single human protein because we haven't used that protocol for every single human protein. However, for the subset of targets that we worked on so far, yes, these universal buffers, a denaturing buffer that's SDS containing for Western blot, because we're working on the denaturing conditions, and a non-denaturing buffer for the iPSA, where you would want to capture the native protein. Yes, these have been successful so far. We worked on cytoplasmic cytosolic proteins, nuclear proteins, lysosomal, and even inflammasome-specific proteins, which is NLRP3 that I have in mind. It's been successful at extracting proteins from, we even worked on mitochondrial proteins as well. So yeah, this broad selection of proteins and the success of these commercial buffers and extracting them, I think gives a solid confidence about the efficiency of these buffers and this protocol.
Great. The next question is asking, so when validating antibody for immunofluorescence, you discarded secreted proteins. Why is that?
So, because we're working in cancer cell lines, you do not expect without, at least without a treatment, to be able to retain enough of the protein inside the cell so you're able to immunostain for that. However, in immunohistochemistry, if the protein is known to be stored in an organ inside the animal, then yes, you can immunostain in immunohistochemistry, and I know that's true for proteins like MITKIND, for example. However, in cell lines and in steady state levels, without knowing how the protein could be blocked and stored inside the cell, we cannot validate in immunofluorescence. If that makes any sense.
Yeah. Our next question is from Maria. Maria's asking, so she says, I really liked the presentation and it's a very necessary initiative in research. I would like
Thank
to
you.
know if antibodies can be sent to you for validation.
The YCharOS mission is now focused on commercially available antibodies that are accessible to everyone. So I assume in her case, it's maybe a custom-made antibody or an in-house antibody that she developed in her lab because she couldn't find maybe a suitable antibody that's commercially available for her. So yeah, right now our focus is on what's accessible to others rather than just selected products that may not be as accessible,
Okay, great, thank you.
if that answers the question.
And then I think we have time for one more question. So the last question is, so it's similar to a question that was already asked. I found a lot of inconsistencies in antibodies when moving from cell models to rat/mouse tissue models in Western blot and in immunoprecipitation. Any way to overcome those?
So we're talking here about like more noise in the different species when switching from.
Yeah, it seems like inconsistencies from model to model. So how to kind of, when you're trying to troubleshoot your Western, where would you start if you're going from one model to another one?
If you have the option, again, try to maybe purchase 3 products that are expected or shown to react with rat, and then compare the level of affinity of each of these antibodies toward rat. Because we think of this that, okay, if it reacts with a human and rat, it may react in an identical manner, but that's not actually true. The level of affinity of the antibody may differ from one species to another. So I would say, again, give yourself multiple options if you have that that are commercially available, and choose which is the best reagent. You can always start with a Western blot if the reagent is suitable for Western blot, and then see how your antibody detects the protein, and then you can move on to other assays.
Yeah, this is kind of like the importance of, trusting your antibody supplier because oftentimes on their site, they might tell you what the immunogen is, and you just want to ensure that, that sequence will line up with whatever tissue or sample you're using. And so that might be one of the issues. So yeah, as Riham mentioned, trying multiple antibodies, or if you're unsure, just reaching out to the company because oftentimes they do validation that's not posted on their website as well. So it's possible that they have tried validating it in your tissue and maybe it didn't work. And so they can kind of save you that hassle of trying to troubleshoot an antibody that's just not going to work in your sample.
Mhm.
All right. So thank you everyone for the amazing questions and thank you again, Riham. With the last few minutes, I just wanted to share a couple more slides. Here it is. All right. So I just wanted to highlight some products that might be interesting for our audience today. So Protein Tech offers a quite impressive and expansive portfolio of primary antibodies, and that continues to expand. We currently have antibodies against over two-thirds of the human proteome, including over 14,000 polyclonal, 10,000 monoclonals, and 8,000 recombinant antibodies that are validated in multiple applications. And just a note about our recombinant antibodies. So we call our recombinant antibodies our UniRab antibodies, and our R&D team actually uses a state-of-the-art genetic engineering platform, which we have coined as our ABCE platform, in order to design unlimited supplies of recombinant antibodies that can detect antigens with extremely high precision. They also offer unparalleled lot-to-lot consistency, which is going to be ideal for the reproducibility of your long-term projects. Our team also understands the importance of antibody validation, so whenever possible, we do validate our antibodies using knockdown knockout models, and we currently have over 7,500 antibodies that are validated. Protein Tech also offers secondary antibodies, including our multi-Rab goat recombinant secondaries. Our multi-Rabs are a multiclonal mixture of precisely engineered recombinant monoclonal antibodies, so they can recognize multiple complementary epitopes on the same IgG. So the idea here was that our team wanted to provide a secondary that has the high sensitivity that you see in polyclonal antibodies with the superior specificity of monoclonals. Our team also noticed that there is very minimal cross-reactivity with off-target species using these secondary antibodies. And because they are recombinants, they offer unlimited supply and high lot-to-lot consistency, which makes them great for reproducibility and your long-term projects. And currently, we have multi-Rabs for rabbit and mouse antibodies that are conjugated to either our correlite dyes, HRP, or poly-HRP. And just a note about our correlite dyes. So these are actually our in-house dyes, and they offer high photostability and performance with a similar spectral profile to what you would see with Alexa floors. I also want to highlight our flexible antibody labeling kits. So sometimes instead of using a secondary, it is more convenient to use, a conjugated primary antibody. However, one might not be commercially available for you. Therefore, we offer these kits that will allow you to conjugate your own antibody in your own lab. And the great thing about our flexible kits is that it is compatible with any primary antibody, so you don't have to use a Protein Tech antibody. It can essentially be an antibody from any sort of supplier. So you really can use, if you have a favorite antibody that you just wish was conjugated, you can definitely use that. The kit also works with any antibody concentration and formulation. So with traditional methods of antibody labeling, you need to generally do some sort of buffer exchange in order to ensure that your antibody is in PBS only, and this can lead to a lot of antibody loss, and it becomes very expensive. However, with our kits, you don't need to worry if your antibody comes in a buffer that has azide or glycerol. No exchange is going to be required, and you can also label just as much as you need for an experiment, or you can scale up if you would like. The protocol is also really fast and easy. So within 10 minutes, you have a conjugated antibody that's going to be ready to use. And the way that it works, so you can kind of see here on the diagram at the bottom, is that you basically have your primary antibody, and then you add in a high affinity flex linker that has the conjugates that are covalently attached to it. And within minutes, that flex linker will bind to the FC portion of your primary antibody. You then add in a flex quencher, which will bind to any of the unbound flex linker and essentially neutralize it. Then what you're really left with is a fully functional conjugated antibody that's ready to use. Because you have pre-conjugated your antibodies, this kit is really great for same species multiplex experiments. And it's also been validated in multiple applications. So you can use it for IF or flow or Western blot. And then we currently offer flexible kits for the conjugation of antibodies against these specificities here shown on the right. And then in these different conjugates. So we have, different fluorophores as well as HRP and biotin. We also recently launched our Flexible 2.0 kits, which are optimized for immunofluorescence. So these kits contain more dyes on the flex linker compared to our initial flexible kits, which makes them ideal for kind of the lower abundance proteins. And lastly, when you are characterizing an antibody, oftentimes Western blot is going to be your initial application that you use. So in addition to antibodies against your protein of interest, Protein Tech also offers accessory products for Western blot, such as loading control antibodies in order to allow you to, properly interpret your blots. We also offer a variety of controls based on sample type and a molecular weight of your protein of interest. So that includes our GAPDH monoclonal antibody, and so our GAPDH monoclonal antibody is actually the top cited GAPDH that's on the market. We also offer three different protein ladders. So we have our regular, broad, and extra broad range ladders. So these ladders can really help you identify proteins between 3 to 310 kilodaltons proteins. And lastly, we also offer our Signal Bright enhanced chemiluminescence substrates, which can detect proteins at femtogram levels with a signal duration of over 12 hours. So this is going to be really ideal for kind of your low abundant targets and your precious samples. So with that, I would just like to thank everyone for attending today's webinar. Thank you again, Riham, for your wonderful talk. It was really great to see, the initiative and to kind of support. And if you do have any questions after this webinar is over, please do reach out to us. You can reach out to Protein Tech at events@ptglab.com, or you can reach out to Riham personally at riham.ayoubi@mcgill.ca. And if you liked today's event and you want to know about any sort of upcoming webinars and events that we have going on, please do follow us on social media because we post about all our upcoming events there. So with that, I will go ahead and end the webinar and thank you again, everyone, for joining.